Hello, and welcome to our show. My name is Lorena Lomelin, joined by my co-host, Phoebe Wang. Phoebe is a student at ASU learning about rare disease right now, and we are happy to have her join us. Today, we are thrilled to have Dr. Virginia Kimonis, head of Kimonis Lab at the University of California, Irvine, here to speak with us about research and a novel treatment. Dr. Kimonis is a nationally renowned expert specializing in genetics and clinical biochemical genetics. She is recognized as a physician of excellence by the Orange County Medical Association. Prior to joining the Children's Hospital of Orange County, Dr. Kimonis attended medical school in the UK at Southampton Medical School, completed residency in pediatrics at Massachusetts General Hospital in Boston, and conducted her fellowship training in clinical and biochemical genetics at the National Institute of Health, Johns Hopkins, and Washington, D.C. Children's Hospital. She is actively working with the NIH-sponsored rare disease clinical research. She speaks fluent Hindi and Greek, and she is the lead researcher for the Lost Enzyme Project, a preclinical study aimed at creating a novel treatment for Beta-Mannosidosis. This is a project Aaron and I co-founded and co-chair along with other families of patients diagnosed with Beta-Mannosidosis in the U.S. Thank you for being here with us today. Hi, Dr. Kimonis. How are you doing today?
I'm doing great. Thank you so much. How about yourself?
I'm doing great. And, thanks for meeting with us and answering some questions. We really appreciate it.
Well, thank you for all the trouble coming here and visiting the lab. We appreciate it.
It's been great. Thank you.
And it's always a big inspiration for our researchers to actually see families with rare diseases.
And we're inspired by them. It makes us want to work harder for our kids.
Thank you.
So to kind of jump in on some of the questions that we received, tell us a little more about yourself and what really inspired you to work in the field of ultra-rare disease.
Thank you. So, my name is Virginia Kimonis. I'm a professor at UC Irvine in the departments of pediatrics, neurology, and pathology. So I've been working with rare diseases for a very long time. I started my life actually as a family practitioner, and then I transitioned to pediatrics. And in pediatrics, we see lots of rare diseases. And I actually was always interested in genetics, but there was no such career when I started my training. And I went to Dubai, and then I really got interested in rare diseases because of the high rate of consanguinity, parents being related to each other. There's a high incidence of rare genetic diseases. So I found them fascinating and taught myself and decided to come to the States to train further in genetics.
Wow. Yeah. I didn't know that about Dubai. Now that you mentioned, you've seen a host of rare diseases. What are some of the rare diseases that you've worked with in the past or in your research?
Yeah, actually, thanks for asking that. One of the earliest disease that I worked on a research level was another lysosomal storage disease called cystinosis. With cystinosis, you get cystine crystals collecting primarily in the kidneys, so they get kidney failure. But it also affects other organs, and I did a study on the thyroid gland being affected, and you get crystals depositing in the eyes, so they have difficulty with vision, many other abnormalities. But other rare diseases that I've worked on are probably the main focus of our lab right now is VCP-associated inclusion body myopathy. The patients also develop Paget bone disease, frontotemporal dementia, ALS, cardiomyopathy. Just one single missense mutation that we found in our lab causes a myriad of different features. And we're working on different aspects of this progressive disorder in which people die early, using small molecule inhibitors because the mutations cause gain of function. And we're also using gene therapy strategies. We're using small molecules, antisense oligonucleotides. So knowing what the defect is, what's causing the disease, helps you address it using different strategies.
Wow, you're working on a lot.
You're working on a lot. And as a result of my muscle interest, our lab has also been focusing on another inclusion body myopathy caused by HSPB8 mutations. This is a heat shock protein. So very important when the cells are stressed. You need heat shock proteins to correct the damage of proteins. And so These group of disorders are called multi-system proteinopathies, and you get deposits in the muscle, in the brain, in the spinal cord, heart of these proteinaceous material. And we also work on another lysosomal storage disease called Pompe disease. So in that you have glycogen collect in the muscle. So there are two types of Pompe disease. In the infantile type, people or the kids die by a year of age. They develop massive cardiomyopathy. They're very floppy babies. They have respiratory issues. Then there's an adult type, which we're mainly focusing on because my patients have adult onset type. And we're working again on ways to knock down the gene that makes the glycogen synthase. We're using antisense oligonucleotides to correct that, and it's proving to be very successful.
So those that you mentioned, are they all in the same lysosomal storage family?
VCP is not technically a lysosomal storage disease, not considered a lysosomal storage disease. However, the lysosomes are affected and autophagy is impaired in that disease.
And what is autophagy?
Autophagy is a way that proteins or anything that's destined for degradation gets engulfed in this membrane-filled chamber, and then degradative processes degrade it. So then protein or carbohydrates get recycled. So it's a very important process and during starvation, the body keeps going by using autophagy as the mechanism.
So Dr. Kimonis, can you explain to our listeners what a lysosomal storage disorder is and why they're so challenging to treat?
So what a lysosome first is, it's an organelle in the cell, in the cytoplasm, in which proteins or sugars or lipids are degraded, and several enzymes are required to degrade these material that then are recycled. And as a result of a deficiency of an enzyme because of a gene mutation, then that material isn't degraded, and the lysosome gets chock full of material that shouldn't be there. And then it ruptures and the cell gets destroyed, and you can get, depending on the organ, you can get muscle weakness. If it's Pompe disease, the glycogen accumulates in muscle. In other diseases, you might get the lipid material or the sugar material accumulating in the brain, and you can get problems with cognitive impairment. You can get seizures. When you look at the brain, you can see white matter changes. It depends where the accumulation of this material is. In cystinosis, you get accumulation in the eyes. In Gaucher disease, the liver is massively enlarged, the spleen is. The nerves can be involved, and it causes pain, or the vessels might get occlusion because of the lipid material in the blood vessels. You can get stroke. So there's a variety of clinical features depending on what material is stored and which organs are implicated in that storage.
Right. Some of those symptoms that you mentioned, we see some of those symptoms in beta-mannosidosis. For our listeners, could you give a better explanation of what beta-mannosidosis is?
Sure. So Beta-Mannosidosis is one of the rarest lysosomal storage diseases that I have worked with. Currently, we follow 4 patients in the US, and there are 2 in the UK that have been put together as a case series of a publication. So Beta-Mannosidosis is caused by deficiency of the Beta-Mannosidase enzyme. So an enzyme is a protein made by a gene. So the gene makes messenger RNA, and the messenger RNA makes protein. And when you get defects in both copies of the MANBA gene, that's the MANBA, you get deficiency of the beta-mannosidase, and therefore this sugar material can't be degraded, and it accumulates in the lysosomes, in the brain, in other organs that I mentioned, and it causes many of the medical problems that are associated with beta-mannosidosis.
So you mentioned it is the rarest disease that you've worked on. Reason it's so rare is it just the number of patients, or is it just the symptoms?
That's a really good question. I believe it's rare because people haven't recognized it, because there's such a variety of clinical features that are associated with this condition. For instance, one of our patients only had hearing loss and some developmental issues. Other patients have had much more severe manifestations like seizures, cognitive, having difficulty walking, not being able to swallow, speak. So just a variety of symptoms. Some kids have a lot of pain because of the occlusion of their nerve fibers by this lipid material.
Yeah, it's definitely a host of different symptoms. So, Dr. Kimonis, you mentioned that you feel that it's underdiagnosed. With that, do you mean that like patients are having symptoms, but doctors just aren't recognizing that it could be some type of genetic disorder, or lysosomal storage disorder, or beta-mannosidosis?
Exactly. Because the features associated with beta-mannosidosis are so varied, every single patient that we have has got a completely different manifestation. I believe that more awareness is needed. And also, as testing is becoming more widely available, including MANBA gene in panels for autism, developmental delays, seizures, deafness particularly, which is one of the most common features, pain, skin problems, white matter changes in the brain. There are so many different panels that MANBA gene could be included in. So as testing becomes more widely available, such as newborn screening, I believe we will identify many more patients with MANBA mutations.
I hope that too. So your team is developing a novel treatment for beta-mannosidosis. Can you tell us how that works?
So currently there is no treatment for beta-mannosidosis. Just symptomatic treatment if they have pain, analgesics, if they have seizures, controlling their seizures, but generally there is no treatment. So we're very lucky that the families actually have pioneered a potential treatment in conjunction with JCR Pharmaceuticals, and they have a J-cargo technology in which the enzyme crosses the blood-brain barrier. So with beta-mannosidosis, the brain is definitely affected. The sugar material is deposited in the brain, and enzyme, if administered intravenously, which it is for many diseases like the ones I treat, in which the brain is not involved that much, you really need the enzyme to penetrate the brain. And so this new technology will allow the enzyme to go where it's needed and help the deterioration in their brain function in terms of cognition, seizure development, white matter changes.
So that's the key. It needs to get past that blood-brain barrier.
Absolutely, yeah. So we're very optimistic. We have just started working with this disease in the lab. The family have been extremely collaborative and extremely cooperative. They have donated their skin samples, which have been converted to fibroblasts. We believe that the fibroblasts are a good model, but the plan is to make them into nerve cells, because ultimately it's the brain that we want to treat. We also were made aware that there was an animal model which no longer is being worked on. The animal model hadn't been studied really for the clinical features of beta-mannosidosis, but it did have the sugar deposits. So we are now breeding the animals in our vivarium at UC Irvine. They're breeding very well. We're hoping that there will be enough animals for us to do our own natural history study, because the work that was published was done many years ago. We want to find out, what are the animals manifesting in terms of behavior features. So we're going to test them with standard tests like the rotarod to look at their balance, their performance, their strength, but also cognitive tests. So there are tests like the novelty toy test, the maze and water maze test, and other tests to see how they perform compared to animals that don't have MANBA. We had one of our lab postdocs actually spend 2 weeks in Japan training on how to study the cells and the animals, all the techniques that are needed to actually evaluate both in vivo and in vitro studies that we hope to conduct with this new enzyme.
Sounds like the research is really moving.
It's really moving. They're very excited. And I have a long interest in lysosomal storage diseases. As I mentioned, one of the first diseases I worked with was lysosomal. And currently we work both clinically and in the lab on other lysosomal storage diseases. So I'm excited that once you have a treatment available, we find that there's a lot more interest in diagnosing these conditions. Now we have newborn screening for many disorders for which there's treatment, like Pompe disease as an example. So that's only expanding, and I'm sure we will identify more patients who could be treated earlier.
Right, maybe one day, like you said, add Beta-Mannosidosis to that newborn screen. Yeah.
Exactly.
I guess, you kind of walked us through the development, that we're working towards for a treatment. How long do you think to maybe get to clinical trials? Do you think it's going to be able to reach that phase?
Well, that's called the valley of death, right? The big gap between doing the preclinical work with animals and cells and actually taking it to the patient. It's a very expensive proposal, and typically you need pharmaceutical companies to invest in the clinical trials because of all the regulatory requirements. So our task in the lab is to prove that the enzyme's working, it's getting into the brain, it's helping the cognitive decline, it's removing all the lysosomal deposits that we know that the animal shows, and also the collection of the oligosaccharides, which are the products from the deficiency. So there are many different things we will be checking to see if the enzyme is effective and if it's safe, and then we have to convince the FDA, and that's a long process, to let us treat patients. And there are many phases of trials, starting with volunteers. First of all, animals, volunteers that are healthy, and then a few patients, and then a larger number of patients. With rare diseases, the FDA gives us a break in terms of the numbers of patients that are needed to prove an efficacy. So therefore, the natural history study that we are encouraging our families to participate in through the CORDS registry is really, really important, because once you've established what the normal rate of progression is, then you can test what the effects of the treatment are to see if you're changing the rate of progression.
So we have Phoebe here, who's actually a student at ASU, and she's actually working on this project, and she had a few questions herself, and I'd like to bring her over to the mic and kind of let her ask you.
Okay, hi, I'm Phoebe. So I have a question for you, Dr. Kimonis, and it's kind of looking far into the future where when this enzyme has already been clinically tested and patients of Beta-Mannosidosis are getting the enzyme treatment. I know there are other clinical trials for right now drugs that help with remyelination, because I know Beta-Mannosidosis comes with demyelination and dysmyelination. So do you think in the future, when they're undergoing that enzyme treatment, there's a possibility where they can undergo drugs like PIPE 307 that's undergoing clinical trial to regain those cognitive abilities that were once lost due to this disease?
Thank you for the education. So it's a really good question. What you're asking is, will there be recovery of some of the brain manifestation, like the hypomyelination that's been reported with Beta-Mannosidosis? It's always difficult to say how much you recover if an organ already is affected, but the goal is to prevent any further deteriorations, because we know it's a progressive disorder. People who didn't develop seizures. People have difficulty walking. They do have cognitive decline. And the hope is that if this works, that you will prevent further decline and maybe some recovery, because kids are very resilient, and we see kids with severe effect from their disease, and they do recover. Yeah. So the hope is that that will be the case.
Yeah, I was reading some articles about like some drugs getting tested that actually regrow the myelin sheath. I thought that was very interesting, and it would probably be very relevant and useful to rehabilitating patients of beta-mannosidosis. And I just kind of wanted to ask what that would look like in the future, if there's hope out there.
So it's very important to know what the defect is, right? You need to know the gene, because a treatment that works for this other condition won't work necessarily for beta-mannosidosis, because the mechanism is different. So in beta-mannosidosis, you're collecting, the sugar, and then we're measuring them in the tissue, in the plasma, in the urine of these oligosaccharides. They're sugar molecules. So what we would need to do is add the beta-mannosidosis enzyme to the body, given perhaps as an intravenous infusion once a week or once every 2 weeks, which will then break down the sugar and reduce the material that's accumulating in the brain and other organs that we know that are affected in beta-mannosidosis.
Okay, thank you for answering.
But that was a tough question. My God, I didn't know about the demyelinating studies. No, but you'll have to send me information about that.
Yes.
Yeah. But I hope my answer was okay with that answer.
Great. No. And we'll do a quick, we'll do an introduction, and
Now it's being like developed for multiple sclerosis. And that's
Oh,
pretty
okay.
similar to beta-mannosidosis, the symptoms and all that. So I was thinking, once you start breaking down those sugars, when you get the enzyme treatment, and then you accompany that with other sort of rehabilitating drugs, I'm wondering what sort of improvement you can see, what potential there is.
a researcher's brain
it's
right
very
there.
hard. It's very hard. That's a very good question, because we have that issue happening right now where we want oligo. We want an antisense oligo and enzyme, because enzyme works great on the heart, antisense oligo works great on the muscle. But these companies don't want to work with each other. They want monopoly on their product. So that is always an issue, and also the cost and the more side effects, because no drug is perfect. Yeah. So I mean, the basic problem with beta-mannosidosis is that it's the enzyme deficiencies. So I think that's the first thing we should correct, right? Just replace it.
So, we mentioned gene therapy earlier. So, can you explain to us, what's the difference between enzyme replacement therapy and gene therapy?
So enzyme replacement therapy is basically giving the protein or the enzyme into the body intravenously. So it's replacing what the body is deficient in. So it works in the target organ, be it the kidney or the liver or the brain or heart. And what gene therapy is, it's a way of introducing the gene. So it's not the protein, it's the gene that makes the protein. So it's added on to a virus, typically an adeno-associated virus, or AAV, and there are different types of AAV, the most common being AAV9. And once it enters, it's given as one infusion, and it could be then lodging in the liver and producing the gene of interest as it replicates, as this virus replicates. So it's typically considered a one-time treatment, and then the virus keeps producing this gene of interest, which then makes the protein of interest. And I believe that is also a very promising therapy, because many of these lysosomal storage diseases, once you penetrate the right organ, you need to get it in the right organ, you only need a little bit of enzyme. A little bit goes a long way for especially the lysosomal storage diseases. Yeah. Generally, there are certain diseases like Pompe disease in which it has to penetrate the muscle, which makes up the bulk of the body. And so you need big amounts of enzyme, for instance, or gene therapy to penetrate the muscle to make a difference in the natural progression of the disease.
So you mentioned these possible treatments that are being worked on in your lab and in others, but those sound expensive. And so we kind of want to talk about the funding side of research. Doing this kind of research, does it take a lot of money? Can you explain to our listeners why it's so expensive?
So rare diseases are rare, and typically the bulk of the funding for research goes to common diseases like cancer, like Alzheimer's disease. So rare diseases have to compete with these much more common disorders. However, we all believe that investing in rare diseases is really important because it helps you understand the biology of disease, and it helps you understand the mechanism for common diseases as well. And there are many instances of treatments for common diseases like hypercholesterolemia, for example, has arisen because of the work they did with these rare syndromic types of those diseases. The work that we do in the lab is very expensive. Working on cell lines, making stem cells are expensive. The supplies that we need to make these stem cells grow is very expensive. Making new targets, new treatments such as gene therapy, the virus, making antisense oligos. They also require a lot of expertise and funding, but also the work that needs to be done in animals before we take it to the patients, because you need to show that the treatment is working. The biomarkers that we use to track the disease is actually improving. And animal work is expensive. It takes a long time, a lot of patience, and very exacting work. So all of this needs funding to prove that the enzyme is not going to harm patients, because you don't want your treatment to make the condition worse than Beta-Mannosidosis already is. So it has to be done with great care and safety.
So thank you again, Dr. Kimonis. I'd like to end with one more question, and more of like a personal one, I guess. Is there any patient stories that have stuck with you throughout your career?
Yeah, my life has been touched by so many patients, and as you know, Beta-Mannosidosis is an ultra-rare condition, and there are only 4 patients that we know of in the U.S., but I've been touched by how much the 4 families have achieved. They have raised money, so the work that we are currently doing in the lab with the cells and with the animals has takes funds, and the families have done an amazing job raising funds, raising awareness, connecting the research lab with a big pharmaceutical company who have been incredibly helpful and collaborative in developing a treatment. So I'm really touched by what the families have accomplished with Beta-Mannosidosis. I'm also touched by some of the successes of treatments. One of my patients texts me regularly because he appears to be cured from his disease, and every time he would have his treatment, he would have terrible pains, and now he feels great with his gene therapy trial that hopefully will be approved for his disease. And patients like that do stand out and are the reason why we need to keep looking for treatments for patients with rare diseases. Even one patient matters because we learn so much from that one patient. Thank you.
Dr. Kimonis, it's been an absolute pleasure speaking with you today. Your work is not only groundbreaking, but also deeply inspiring, and it's been offering hope to so many who previously have had none. So I just want to personally thank you and everyone in your lab that are working towards not only Beta-Mannosidosis, but all of, a large spectrum of rare disease. And I hope that this motivates others and other researchers and other pharmaceutical companies to really see that there is benefit in our ultra rare disease community. So thank you, and happy holidays.
And also the families, I commend you to continue the good work with raising awareness and lobbying for extra funding for rare disease research.
Yes, we're going to be out there lobbying. Thank you for joining us on These Kids Can't Wait. Every story shared brings us one step closer to awareness, advocacy, and action. If you've been inspired, please subscribe, leave a review, and share this episode.
To learn more about the Lost Enzyme Project or donate to the cause, you can visit our website, Instagram, or Facebook page. Those links will be provided in the show notes below. Thank you again, and remember, these kids can't wait, but together, we can make a difference.